Thank Professor Lee for kind introduction, and thank organizer and Makrojén for giving me this honor. I believe this community want me to do work more harder for this KSMCB, and as well as the general scientific community, and I promise I will do that. Okay. In this seminar, I will shortly introduce structural biology, and I will show the previous study on the microRNA biogenesis. And main topic is the current studies on gap junction. And if I have time, then I will shortly show my contribution to the gene editing field. So we can study biology in many discrete levels, and structural biology I'm sorry, and structural biology to deal with the smallest level. And we solve the structure of biological macromolecule and clarify the molecular mechanism. And this field require high-end technology such as X-ray crystallography, NMR, and electron microscopy. And electron microscopy is advanced so much, and now with cryo-EM, we can solve the supramolecular complexes and membrane proteins much more efficiently than other methods. And electron tomography is quickly advancing. So I believe in near future we can study molecular society in cells. And this tool is not only for the structural biology, it will be for the cellular and tissue biology. And there is another important thing required for the current structural biology. It's pure protein. During my PhD and postdoc and ETH jury, I studied many proteins in many different fields, but I failed most of them because I could not get the pure protein. We use usually equal expression system, but only a small portion of human proteins can be expressed by this system. And insect yeast cell system didn't really help much, so I started developing the human cell expression system for higher expression and lower cost labor, which is necessary for the structural biology. And I spent most of the time in ETH jury for this development, and I finally got the ABCA1 membrane protein, but this protein did not crystallize, was not crystallized. But the system, however, was very useful for the next project. I moved to the Center for RNA Research, where Professor Larry Kim is the director. And from 2013, I started my own group, small group, and I collaborated with two talented postdocs from Larry's group, and we focused on the initial processing step of microRNA biogenesis. In 2001 to 2003, Larry already found primary microRNA transcript, and its processing factor DROSHA and its cofactor DGCL8. This complex is called the microprocessor. But it was a mystery how microprocessor precisely cut the hundreds of different prime microRNA. It is important because this cleavage determines the mature microRNA sequence. So with the human cell expression system that I developed and the rational design of protein constructs, we could purify and reconstruct the various complexes. And we performed extensive processing assays for various substrates, and we could dissect the functions of DROSA and DGCR8. And the minimally functional complex was luckily crystallized, so we could solve the structure too. So the previous wrong model was corrected by our biochemical study, and this was much more improved by our structural study. But with this study, we still could not completely understand how microprocessor precisely cut one single site. So we focused on the interaction between the GHG and DSRBD, and we designed the high throughput processing assay. And we determined the rule of cleavage site selection. These are prime microRNA features, and DROSA measured the distance from the basal motif and GHG motif, and DGCR8 measured from apical motif. And all these motifs contribute, but there is a significant hierarchy between these motifs. If this position contains optimal sequences such as GCG or CCG, then this motif dictates the cleavage site selection. However, the most prime microRNA do not have these sequences. Then microprocessor consider other motif for the cleavage site selection. So this rule explains most of primary microRNA processing. So in 2017, I moved to Korea University and started working on the gap junction. And this field also has a long-standing question of how this channel open or close. Because gap junction is a membrane protein, and cryo-EM is much better tool for the membrane protein than the X-ray crystallography. So we changed the main tool from the X-ray to the electron. There was only one cryo-EM machine in Korea at KBSI, and Jaekyung and Hyungsub was setting up this machine. So we started our collaboration from 2015, and I provided better samples every time. So we failed many times, but finally, 2018, we solved the first cryo-EM structure in my lab. And it was the gap junction hemi-channel, and the resolution was very high. This was actually the best resolution for the membrane protein by cryo-EM at that time, until 2020. But I will not talk about this story today. So I brought more recent research on the gap junction. So before I show you the structure, and I will give a short introduction of a gap junction. Gap junction is a unique intercellular channel. It forms the direct passage between the two cells, and molecules smaller than 1.5 kilodalton can move through. Connections are the subunits. The connections are expressed, and they are oligomerized in ER and transported into the membrane. And this hemi-channel function as itself is sometimes open to release ATP and glutamate for inflammation or neurotransmission. But usually they form the dock together between the cell and form a gap junction intercellular channel. They are usually open, and their main function is metabolic synchronizing. importantly, these channels are regulated by many factors. And the biggest advantage for the gap junction is its quickness. Once the signal is reached one cell, that it is quickly synchronized. So this is crucial for the heart beating. And CX43 member connect all the cells in the heart muscle. And it's also important for the nervous system. We know very well chemical synapse. However, there is electrical synapses connected by gap junction channel. And chemical and electrical synapses are essential collaborators in animal neural circuit. And the structure was solved in 2009 already, but it showed that about 10 angstrom pore. So they believe this is open state, but gap junction, more bigger molecules than bigger than 10 angstrom. These molecules can also go through the channel, so it is still unclear if it is partially open or fully open state. And also there is no structure of the closed state. So we focused on the CX43 member, and because it is expressed in most cell and most studied, and it's also relevant to heart disease and cancer. And electron crystallography structures was solved already in the 1999, but this low resolution structures was not improved so much so far. We recently solved this structure of CX43 channel, CX43 channel, and this showed the structure showed a significant difference from the previous structures, especially in here the N-terminal helixes are collocated at the gate, and this formed the smaller pore size than the previous one. We called this conformation the gate covering NTH conformation, shortly GCN conformation. And this structure was solved at pH 8, one of the channel closing conditions. And we also solved the structures in the channel opening conditions, for example, pH 6.9 or deletion with the mutant with the deletion of C-terminal loop. This structure showed the pore lining NTH as well as a gate covering NTH, and also mixed state. Actually, most of the particles contain both conformations. So we think this NTH conformational change is individual and random, and we captured in a cryo-EM samples. So we captured the equilibrium state. And we also inspected the surface charge and the pore at the pore in fully GCN conformation. The pore shows the positively charged the surface, and fully PLN showed the negatively charged. So these two channels, the conformation has ion selectivity, the chloride selectivity, and sodium selectivity. But mixed conformation do not have charge selectivity. So this is consistent with the previous physiological data. So we believe this is probably open state. And we also inspected the pore size in 3 different conformations. and mixed conformation show the bigger pore, which the NAD plus or fluorescence dye, the bigger than ATP, can go through this pore. So we think this is also fully open state for the large molecule. Since we solved two different conformations at high resolution, so we could compare between the two conformations in detail. And we found an interesting structural change in TM1. And it was an alpha helix to phi helix transition. Once in the middle of TM1, because it changed in the middle of TM, half of other part should turn around about 50 degrees. I will show you the movie showing. So the yellow part is the alpha to pi helix change is happening. So you can see half of helix is rotating. So the international network between the helixes totally change. This change also make a small gap between the two subunits. So you can see the subunit pore here. So this gap is located in the inner leaflet. So the lipid molecule probably can go in and out from the inner leaflet. So we made this hypothetical gating mechanism, as there are still many questions that we have to solve. Here the open state, we believe this fully open state is probably mixed state, and it does not have no charge selectivity. It does not have charge selectivity, and it is ATP permeable. And probably the completely closed state, the lipid can go in through the side opening, and so occluded form may be the closed state. So I think I have some time to show my contribution to the gene editing field. Now, I collaborate with Professor Sang-Soo from 2015. We initially started this collaboration to develop the human cell expression system further, but we quickly expanded our collaboration to develop a safe base editing method. As many of you know, the classical CRISPR-Cas system is proven not appropriate for the medical use because its complete cut of chromosome can cause a large chromosomal deletion. The base editor, however, can be much safer because CRISPR-Cas system only basically recognized the sequence, the target sequence, and the fusion protein deaminase modify only a single base. An adenine base editor is the current best one for medical use, but it has a significant problem, or off-target problem, which is the cytidine deamination activity at TC motif. And it is also difficult to obtain pure protein. So with the rational design of mutations based on the structure, we modified this adenosine deaminase to have the precise, the AB without C editing activity. And we also luckily found TC specific C editor with little A editing activity. Yeah. And we also developed the RNP delivery method with purified ABE. We could purify the AB protein in high purity from the human cell expression system, and ABs live only 1 day in RNA transfected cell, compared with the plasmid transfections had a longer time, like a week. And this system decreases the off-target effect very much. So with both technology, we are trying to develop the therapeutics for human disease. So I thank all my group members first, and intimate collaborators, and also I thank Seokyeongbaek Foundation and National Research Foundation for supporting our work. And thank you for your attention.
Thank you for Knight's presentation. And his research covers various kinds of biomedical science, including human microprocessor and intercellular channel and CRISPR-Cas9. I would like to receive a comment or question from the floor. Are there any comment or question? Yes. Jae-sung, I have one question. I said,
Yes.
Intracellular channel is originally membrane protein. Membrane protein.
Yes.
Yes, yes. It has been known that it is difficult to crystallize and identify the structure of membrane protein. My question is, what is your know-how or any scientific tips to crystallization of membrane protein? And do you have any your know-how?
Sorry, I cannot hear very well from here.
So it's okay. Yes, my question is the membrane protein.
Yes.
Membrane protein is very difficult to crystallize.
The cryo-EM, with cryo-EM, we don't have to crystallize the protein. So the membrane protein is extremely difficult to crystallize. So we changed the tool to the cryo-EM. Cryo-EM do not need crystal.
Yes,
Yeah.
it's cryo-EM.
Yes.
Cryo-EM is the overcome the hurdle for the membrane protein.
Yes.
Oh, yes.
Yes. But there is a hurdle for the protein purification also. But the cryo-EM also use much smaller amount of protein than X-ray crystallography. So cryo-EM actually reduced the hurdle both for the crystallization and the purification of the protein. Yeah.
Okay. If cryo-EM is recently is a hot topics, and for student or for audience, and you can emphasize the advantage or strength of cryo-EM. Is any other things for cryo-EM? What is the strength of cryo-EM?
What is the
strength, advantage of cryo-EM?
Cryo-EM? Yep.
Yes.
The question was what is can you say that question again? What? I'm sorry.
Okay. Okay.
You mean
yes,
that just generally for the cryo-EM?
Okay, so there is one question from the floor. Okay.
It is very nice talk, and might be this is going to be really naive question, but I want to ask that one. So when you actually figuring out your structure, the plasma membrane protein structure, you did that one when they actually already inserted into the plasma membrane, right? So I'm wondering that have you considered to check their structure during the delivery time to the plasma membrane? And if you can do that, there is a possibility that might be difference between the during the delivery time, the structure on the delivery vesicle, whatever, and the final form at the plasma membrane.
Yes.
So yeah.
I can tell you that. So we use the detergent most of the time, but we also solve the structure in the lipid nanodisk. It's a lipid environment, and the structure show the similarity. Yeah, not exactly the same. That there are different composition of different ratio of the GCN and PLN conformation, but this is also in vitro system, and we have to choose lipid molecules are extremely various, and we actually cannot really see the structure in real membrane so far. So there is always questions, because we also know this structure is modified. I mean, this conformation can be regulated by the lipid molecule like cholesterol. So if we add more cholesterol, then maybe the structure change to the other conformation. So this kind of problem, I think, it will be difficult to solve by the current technology. But this is why the cryo-electron tomography is so important. So I actually have a plan to move to the cryo-ET technology. So it will be difficult, and I still the technology is advancing. So we have to solve this problem completely. We can solve this problem completely by the electron tomography. Then we can just solve the structure from the real cell and real cell membrane.
Right. Yep. Thank you.
Thank you.
Are there any comments or questions? Okay, one more thing. Just come here. Come to the microphone.
Thank you very much for your inspiring lecture. It can be a little broad question, but I was wondering, so there have been some recent reports on machine learning-based structure prediction methods such as AlphaFold. And what is your insight of how structural biology can benefit from these prediction methods? Because of course these prediction methods are not 100% correct.
I'm sorry, I could not understand.
So briefly, how do you think structural biology can benefit from structure prediction methods using machine learning?
I'm really sorry. I cannot hear very well in the oratorium because of this the sound that maybe we can. I mean, privately we can, I could probably understand much better, but I'm really sorry for the Professor Lee.
After session let's please communicate personally in the floor.
Yeah. Okay.
And
thank
the
you.
question is about machine learning tools. Your insight for recently machine learning is
AI. Okay,
yes,
I
AI.
see.
Everybody is
I see.
expected that the AI machine learning is uncovered the whole structure of protein.
Okay.
Now, what is your insight?
Yes. We actually tested it, the AlphaFold for this structure. So it could solve the structure roughly the same, but the conformation change it cannot predict. And one of the two conformation is not even in the candidate. You know. So I believe the AlphaFold may solve the structure of very rigid protein, and also if there is many sequences, which means it's a highly conserved protein from the bacteria to the protein, maybe AI can solve the structure. It is basically based on the evolutionary interaction between evolutionary conservation of the interaction between the amino acid. It's a co-evolution. So I think cryo-EM now it's solving the actually dynamic structures. It can solve the many different conformations in one sample. So I think the AI cannot really. I'm not sure how much it will advance, but in the current stage, the structural biology still have to do. I mean, the experiment have to do solve the right structures, and also the conformation changes should be confirmed by the experiment, and also cryo-ET will probably show the molecular society and many other complex, and it's still difficult. AI can predict very low affinity interaction. So if it is strong interaction, may be possible, but if it is, I mean, in biology, low affinity interactions are probably more important. That they are more dynamic, and we are always trying to solve the problem in the lower interaction. So I think structural biology still has many things to do. Will be, I think, more things to do. Yeah.
Okay, thank you very much.
Yes, thank you for your questions. For time limitation, I would like to close this lecture. And thank you for Dr. Wu's lecture. And I would like to close this lecture with the floor.
Thank you very much. Thank you.